This is question 2, a 6-mark question from paper 2 of the AS Biology 2017. And this question relates to a practical that you will have done in AS. And it starts by asking, the cells of beetroot contain a red pigment. We know that is betulin. And a student investigated the effect of temperature on the loss of red pigment from beetroot. Now, when we did that, we did it with temperature and we did it with alcohol. He put the discs cut from the beetroot into tubes containing water. He maintained each tube at a different temperature. After 25 minutes, he measured the percentage of light passing through the water in each tube, and we did that with a colorimeter. The student put the same volume of water in each tube. And the question asks, explain why it was important that he controlled this experimental variable, i.e. putting the same volume of water in each tube. It's really important that you stop the video now and have a go at that question to see whether you would get both marks, because only 3% of students actually did. So surprise yourself, stop the video and think about the question in a practical sense. Okay, I'm going to reword the question. The question's basically saying, why can't we just pour any volume of water on the discs? And if the question is rephrased like that, then the answer is quite obviously that we wouldn't be able to compare the result at different temperatures. Most students got the mark to say that if too much water was placed on the beetroot, then the pigment would be diluted more, it would be less concentrated. But very few went on to say that we need to control it because we need to compare the results for different temperatures. And if you've added different volumes of water, you won't be able to do that. That's why sometimes I think if you just think about it practically in a common sense way, think about you being in the lab and doing the actual practical, then the answer comes to you in a much clearer way. Okay, looking at the next part of the question, it says, describe a method the student could have used to monitor the temperature of the water in each tube. Now, it's an interesting question, this, and we certainly don't want to get caught out by it again. Only 17% of students got this mark. And it comes down to the word monitor. If you're monitoring something, you're checking it more than once. And that's what you needed to say. In answer to the question, we just needed to say, take readings during the experiment using a digital thermometer. Now, obviously, most students just wrote, use a thermometer or a digital thermometer, or a digital sensor or whatever. But the actual point was to say that you were taking the measurement more than once. So the word readings, i.e. plural, infers more than one, and during the experiment infers over a period of time. How ridiculous to lose marks on an A-level due to the word monitor, due to something so simple as that. And I agree, it's ridiculous really that you would be losing marks for biology on that because it doesn't really test you on your biology knowledge or your understanding of concepts. It's just testing you on your English language really. But either way, let's not get caught out by it again. When we see the word monitor, we know we mean checking it more than once. The next part of the question gives us some points plotted on a graph. Thank you, very nice, very easy. And it just asks us to draw a suitable curve. Now, we can either do a curve of best fit, otherwise called a line of best fit, or in biology we can do dot to dot unless we're told otherwise. So the safest thing to do is to go dot to dot. And that is because in biology we can't always predict the intermediate values. So that's why we are able to go dot to dot unless told otherwise. Now that graph actually is screaming out that it is a curve. And it might surprise you to know that over 50% of students lost the mark for that because what they did, they weren't careful enough and they extended beyond the plotted points. So don't extrapolate beyond your data values. Last and final part of the question says the decrease in the percentage of light passing through the water between 25 and 60 degrees is caused by the release of the red pigment from cells of the beetroot. Suggest, and that means we're not meant to know the answer, we're meant to be able to work it out, how the increase in temperature of the water caused the release of the red pigment. So all we needed to say was that this was caused by damage to the cell membrane, and the proteins in the membrane will have denatured. And of course, for greater understanding, we know that proteins are in the cell membrane. One of the things that they do is that they make the phospholipid bilayer more stable. So with them denatured, then the phospholipid bilayer would increase in fluidity. But it was enough just to say that the cell membrane would be damaged and that proteins in the membrane would denature, and that would cause pigment leakage. Now, according to the examiner's report, a lot of students wrote about the cell wall bursting. Well, by the time the cell wall bursts, the effect of the cell membrane is negligible because the cell membrane would have been completely destroyed by then. So this has nothing to do with the cell wall bursting. Remember that the cell wall actually has big gaps between the cellulose chains, so it doesn't discriminate what can go in and what can go out. Only the cell membrane does that. I hope the video has been of use to you, and thank you for watching, and please do leave me a comment.